Well, hi, everybody. It's really my great privilege to introduce our last speaker of ViDA 2021, Dr. Roy Wise. I know Roy from a long time ago because when I had my first experience in research as an undergrad at Concordia University in Montreal, that was at the end of the 1990s, so a long time ago. Roy was then the director of the Center for Studies in Behavioral Neurobiology, the CSBN, at Concordia, which he directed between 1983 and 1991. After which he moved on to the NIDA, so National Institute of Drug Abuse, as chief of the Behavioral Neuroscience section branch in 1997. So he lives in the United States, but he often comes to Montreal, where I think he left a part of his heart. So there may be some younger people here today that do not know Roy very well, but I think many others are well aware of the immense influence that Roy and his team have had on the field of dopamine research over the past decades. His CV lists over 300 publications. He published multiple papers over the years on many topics related to dopamine and drug addiction, but he is perhaps most well known for his influential theories and experimental work on the contributions of dopamine to positive reward and to the effects of multiple drugs of abuse, including heroin and cocaine. As an example of his influence, I mean, many generations of scientists in drug addiction have been influenced by papers published by Roy, such as this major review called A Psychomotor Stimulant Theory of Addiction, published by Roy and Mike Bozard in 1987 in Psychological Review. This is a paper that was cited over 3,000 times. Roy's work has been globally cited more than 50,000 times over the years, and he has an H index of over 100. So showing how big an influence he's had on multiple generations of scientists. So Roy's talk today is entitled A Dopamine History. And this is a great way to end the present virtual dopamine meeting. So I'm hoping that Roy and all of you actually join us in Montreal in May 2022 for the next in-person international dopamine meeting. So Roy, the floor is yours.
Okay, thank you. I want to thank the organizers of this meeting again for two reasons. One is they did this last year and they've done it again. So congratulations, you've done a great job. Okay, a history of dopamine. So that was my assignment, and I have to tell you, I couldn't get it all in. I have 11,641 references in my EndNote file, and I had to narrow it down to about 52. So I'll get going. I'm going to stop my talk in 2004 when this movie came out. Some of you weren't alive when this movie came out, and if you were alive, you didn't see it, and you can't get it anymore. I don't recommend it anyway. I'm going to start with the discovery that this molecule had some pathomimetic actions made by Sir Henry Dale in 1910. He only named the compound 40 years later. Peter Holtz discovered dopamine decarboxylase in 1938. Blaschko identified dopamine as intermediate in the synthesis of noradrenaline, but he later suggested that it might have a function of its own, as it turns out. Catherine Montague identified dopamine in the human brain, adrenergic consequences, and they were independent of the dopamine metabolites noradrenaline or adrenaline, starting to suggest again a function of its own. Von Euler showed that dopamine in the lung is 100 times more prevalent than noradrenaline and is distributed independent of noradrenaline. But it was Arvid Carlsson who was really the master pharmacologist in the catecholamine field. He had a postdoctoral fellowship with Bernard Brodie in 1955. He was there for 5 months before he returned to Sweden and began working with his colleague, Nils Åke Hillarp. He decided, Brodie had found that reserpine depleted serotonin, and he thought that this was going to explain the sedative effects of reserpine. Carlsson went back and decided to look at catecholamines, which are also depleted by reserpine, and in fact, he was right. They first reported that L-DOPA could reverse the bradykinesia caused by reserpine. They used L-DOPA because it crossed the blood-brain barrier and dopamine did not. Based largely on findings of Bertler and Rosengren, who showed its abundance in the striatum and suggested that it might have an independent motor function, Carlson was the first to suggest that L-DOPA would reverse the symptoms of Parkinson's disease. And at the time he suggested this, many leading scientists disagreed. In Canada, Cirques showed that L-DOPA could elevate dopamine levels in the rat. Poirier subsequently established an animal model of Parkinson's disease by making lesions of the substantia nigra. And Andre Barbeau with Cirques again found that decreased dopamine excretion in the urine of Parkinsonian patients and showed that L-DOPA could reverse the symptoms of Parkinson's disease. Hornikowitz in Austria had shown dopamine depletion in the striatum of Parkinsonian patients, and with Werkmeyer showed the anti-akinesia effects of L-DOPA. This is 1961. It was 6 years later that Casillas finally found a system that worked, had Parkinsonian symptoms reversed using L-DOPA. He used L-DOPA at 100 times higher doses than had been used before, and he gave it in repeated application, and that reversed the symptoms of Parkinson's disease. Parkinson's disease was defined by Parkinson in 1817. Involuntary tremulous motion, lessened muscular power, propensity to bend the trunk forward, pass from walking pace to a running pace, and this in patients with intact senses and intellect. These were patients like Muhammad Ali, Michael Fox, and Alan Alda that you're familiar with, patients who slowly developed the symptoms of Parkinson's disease over a period of decades. But when I was, shortly after being a graduate student, I first learned about Parkinson's disease by reading Oliver Sacks' book, Awakenings. It was about post-encephalitic patients that had Parkinsonian symptoms and were treated by L-DOPA. And L-DOPA made them completely recover. So these were paralyzed patients with muscular rigidity. Some of them couldn't talk. They were given L-DOPA and they burst onto the stage. They could dance, they could eat. Unfortunately, there are limiting side effects that you all note about. These patients were in the full late stages of Parkinson's disease, and that was kind of the picture I had of Parkinson's disease for quite a while. Okay, I want to talk about some of the fundamentals historically. First, the histology. Irvin Ungerstedt did a beautiful set of thesis projects published in 1971. He mapped the pathways. He used the Falk-Hillarp method, which had been developed by Falk in Hillarp's lab, and that Dahlstrom and Fuchs had already used to identify the cells and the terminal fields. But it didn't have the sensitivity to identify the pathways, which Ungerstedt did, and he did it by making knife cuts. So if you make a knife cut here, the cell bodies synthesize dopamine, they pump it towards the terminals, it gets to the knife cut and it's stuck. But it gets enriched in this little part of the pathway, so you can trace this little part of the pathway. And then in the next animal, you make a knife cut a little further, you can trace the pathway a little further and a little further, and pretty soon you can trace the whole pathway. Lindvall and Bjorklund developed the glyoxylic acid method a few years later to identify fiber pathways directly. You could actually see these fibers with the glyoxylic acid method. There was nothing like injecting a fluorophore into them and just looking. They identified a mesocortical pathway that innervated the cortex using the glyoxylic acid method. As you've learned this week, the major histological issue now involves differentiating subtypes of dopamine neurons and differentiating their afferents and differentiating their efferents. each of which makes a difference. The classic electrophysiology, many authors have talked about this, but I'm going to repeat it, was done by Tony Grace in Bunny's lab. He identified two kinds of firing of dopamine neurons. One was pacemaker firing, tick, 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 tick. The cells could fire quickly, or they could fire slowly, and that's modulated by GABA. So GABA makes the cells fire slowly. GABA antagonists disinhibit the cells and allow them to fire faster. He also identified burst firing of dopamine neurons. Here, the cells fire in bursts that are linked to each other. And this bursting pattern is triggered by glutamate. Bunny found it by ejecting glutamate ionophoretically in anesthetized animals or putting it in a bath where the afferents to the cells were disconnected. And Grace suggested several years later that there were separate functions for the two firing patterns. This firing pattern controlled one thing, and this firing pattern controlled something else. And I'm repeating his work because he was correct. And this is not fully recognized by people, even people in the field today. The neurochemistry. Neurochemistry was done in two ways. Mark Whiteman, working in Ralph Adams' lab, where did he go? In the 1970s, has been working on voltammetry over 50 years, and there are very recent papers that you can find in last year's topics. The method is sensitive to detect dopamine down to a concentration of about 20 nanomolar. This is much higher than the resting potential in the freely moving animal. He developed fast scan cyclic voltammetry, which people in this century have been using almost exclusively. And it detects the effects of dopamine release from burst firing neurons. Burst firing neurons show an accumulation of dopamine that results from several spikes all in a row. The original thought was that it fires before it decays, it fires again, before it decays, it fires again, and that causes these peaks. We now think that maybe the consistent stimulation is more important for blocking the dopamine transporter than for causing repeated firing, because not a lot of dopamine comes out from successive pulses in a burst. How did he do it? He did it first with small probes, 5-micron probes. You can put them in the brain, jiggle them around until you got a good dopamine response, which suggests that the release sites for these responses were anatomically distributed in the space. He used fast cycle time so that he could see the accumulation over the period of bursting. And he used something called background subtraction. The reason for background subtraction is that the electrochemical signal that he's using to identify dopamine slowly decays over time. There's electrical changes. It's also sensitive to fluctuations in pH. So you don't know that at this point in time or at this point in time, you're not sure that what you're looking at is dopamine. But you can find out what's going on here. You generate voltammograms, which are oxidation reduction currents. They look like this, and they're taken at the peak of these spikes. So fast scan cyclic voltammetry measures dopamine peaks, and it measures only dopamine peaks. By background subtraction, he eliminates any fluctuations due to the baseline firing of the cells. This is a problem for fiber photometry too, because the intensity of fluorescence is concentration dependent, and what you tend to see is the peaks and have trouble discriminating what background firing looks like. Craig and Rice have shown that dopamine doesn't travel very far from the release site, 4 or 5 microns. Again, this suggests that the spikes from these peaks are anatomically distributed. They're distributed in space, and they're also also distributed in time. I'm not sure that fast scan cyclic voltammetry detects much of baseline firing. Certainly detects the peaks. The other method of neurochemistry was microdialysis. This was Urban Ungerstedt's idea. He said, imagine a blood vessel in the brain. Ions moving through the cell to be collected, so they pump fluid into the brain, they extract fluid from the brain, and they do the electrochemistry in an instrument. And with this technique, you can measure dopamine as low as 0.1 nanomolar in animals that are treated with dopamine on cell bodies or with tetrodotoxin. So microdialysis measures pacemaker firing, at least in resting animals that aren't being seduced into activity by surrounding stimuli. John Hebbian and Paul Greengard identified dopamine as a slow synaptic transmitter, acting through G proteins and adenyl cyclase, and also identified dopamine receptor subtypes, particularly differentiating between D1 and D2 receptors. And Richfield, Penny, and Young showed that D1 receptor operates at low affinity, while the D2 receptor operates at high affinity. And I put this in fine print because, again, this is a speculation that I've been making for about 10 years, that if the receptors are all close to the release site, that would suggest that the D2 receptors are more occupied by dopamine than the D1 receptors. And that would make the D2 receptor sensitive to inhibition of dopamine, to inhibition of firing of dopamine neurons, but insensitive to elevations of dopamine. And it would make the D1 receptor more sensitive to dopamine released. And Sabatini confirmed that in his talk yesterday. Okay, I'm going to turn to 3 topics of behavior. These are my preferred topics. I want you to take notes. There's going to be a test. Here's the first one. Animals lacking dopamine, animals without dopamine, lack learned behaviors. They have reflexes and instincts, but they don't learn anything. They don't learn anything for one reason, because they're all hunched up, sitting stationary, not moving. But they also don't learn anything because dopamine is the main transmitter for helping to stamp in long-term potentiation. How do we know this? Well, the clearest findings are from Richard Palmer's lab. They made mice lacking tyrosine hydroxylase, the dopamine precursor. And these mice are hypoactive, they're aphagic, they're adipsic. They have to be artificially fed after weaning. They usually did it by giving L-DOPA injections twice a day. With twice-a-day injections, they could, the rats would maintain normal body weight. The depleted rats, excuse me again, mice, had reflexes. They can swallow food if you put it in their mouth, and they can escape from pain if you stimulate them. But what they fail to do is to seek food, and they fail to avoid predictable pain. So dopamine-free mice don't learn to search for rewards. They don't learn to respond to the stimuli that predict what rewards are going to be. And they don't learn to avoid punishment by responding to stimuli that anticipate coming punishment. Palmer's group also showed that feeding can be restored by replacing tyrosine hydroxylase in the dorsal striatum, and that locomotion can be restored by replacing tyrosine hydroxylase in the ventral striatum. This work all confirmed earlier work by Ungerstedt, where he lesioned rats in adult rats. Again, showing that lesioned rats were akinetic, failed to eat, failed to drink. They can swallow food, they can put it in their mouth, but they don't learn to search for it. They can escape from irritation or pain, but they don't learn to avoid it. So again, the animals have to be artificially fed if they're to be kept alive. And I remember, can't remember his name right now, but telling me that they used to tube feed animals twice a day for 6 weeks. during periods trying to see if there was going to be recovery from these lesions. So the bottom line here is that dopamine depleted rats fail to respond to predictors of reward, which tell an animal where to search to get the reward, or predictors of punishment, which tell him whether he should stay put or run away in order to avoid punishment. So there are two forms of behavior that I want to discuss here. Sherrington talked about them in 1906, but I want to talk about Wallace Craig's characterization. He was an ethologist, one of the founders of ethology, and he differentiated two behaviors, appetitive behaviors and consumatory behaviors. So consumatory behaviors mean not just consumption, which is the consumatory behavior for eating, but consummation, which the termination of a series of goal-directed behavior. So in the case of hunger, it's swallowing. Consumatory behaviors are unconditioned reflexes. They're species typical. They're biologically primitive. And they're responses to actual rewards and punishers. Appetitive behaviors, on the other hand, are states of action that terminate when a consumatory stimulus, a reward or a punisher, is actually reached. In the case of feeding, it's searching for food. Appetitive behaviors are learned behaviors. They're responses to external stimuli. These are stimuli that will guide the animal through space to find food in the future. And they control behavior in an interested animal to its goal. I want to pinpoint that, because these behaviors, an animal can be sitting on food and be unresponsive to the food if he's not hungry. But if he is hungry, he will be responsive to the food. So the food is capable of controlling consumatory responses, but it doesn't control searching for them. Consumatory behaviors are reflexes. Appetitive behaviors are learned responses. So what's an animal capable of learning? Ted Hall did beautiful work on feeding in the 70s, 80s, and 90s, and then he started making fancy devices. He showed that rats can learn to eat on the first day of birth. This is a 1-day-old rat pup. He's already been trained earlier today, so I'm going to show you learned behavior in the 1-day-old rat pup. It's going to be a little movie. He's in a cup. There's a lever in the cup. It's covered by cotton batting. There's another lever. If he presses this one, a green light goes on, and he gets a milk injection into his mouth. Milk comes in through this little tube. You can barely see it here. Comes down to. If he presses this lever, a red light goes on, he doesn't get anything. So here's what happens when he wakes up. He presses the lever, he gets the green light, he gets an infusion of milk through this little tube that's running down here out of focus. He falls over in ecstasy, presses the lever again. So normal rats can learn to seek rewards on day 1 after birth. Dopamine deficient animals don't learn to do that ever in their lifetime, and they die of starvation if they're not artificially fed. I want to make a couple of notes here. The first one is that noradrenaline and acetylcholine can establish learned behaviors, but in food-deprived neonatal animals, they don't do anything. They're all hunched up, they don't move, and they don't learn anything. Note 2, Palmer's group has shown that dopamine deficient mice can learn. And in light letters, my inference here, to some degree. They can learn a little bit, and they learn a little bit if you pre-treat them with caffeine, which is logical. It's a locomotor stimulant, and it gets them to move. The trouble with caffeine is that it acts at A1D1 heteromers. It acts at A2D2 heteromers. It acts through G proteins. And so the fact that he had to use caffeine to get these learned behaviors suggests that caffeine itself might be doing some of the things that dopamine does if they're acting together. Okay, so animals without dopamine lack learned behaviors. Item number 2, if you're making notes of my 3 headings, burst firing stamps in reinforcement. Sorry, I'll get this. Okay, so we have a lot of very clever experiments done by many of them in my lab, using dopamine antagonists to block or attenuate the rewarding effects of food and other rewards. This is an example of lever pressing for food. I have a reference list at the end of this talk. You're welcome to look at this if you want to, but I'm not going to walk you through it. This is a problem. Behind my screen here is a picture that you probably can't see. Oh, what a shame. Anyway, he looks just like me.
We do see him, Roy.
Huh? You can see him. Okay, beautiful. He's not Roy Wise, he's James Taylor. He gets his good looks from me. Okay, here's another one. Dopamine antagonist block or attenuate the rewarding effects of food. Again, it's a beautiful study. I suggest you read it. The references are in my reference list. Here's another study by Aaron Attenberg, and I am going to explain this one a little bit. Here the animals run in a straight 6-foot straight alley runway once a day, twice a day, and at the end of it, they get an intravenous heroin reward. For the first 7 days, they run twice a day, and they get a heroin reward twice a day, once in the morning, once in the afternoon. On day 8, they start getting discrimination training. So there's one stimulus that tells an animal when are the trials one a day, when the animal is going to get rewarded, and another stimulus that tells the animals when are the trials where the animal will not get rewarded. Boy, I've got a lot of stuff hidden over here behind the screen. So at one day during this test, the animals are given haloperidol, a dopamine antagonist, and it has a big effect on the behavior. But the critical thing is that the day the animals get the haloperidol is not this day. It's day 15. I can't see that part of my screen anymore. On day 15, they get the haloperidol. On day 15, they want the heroin injection and they run for it. So dopamine antagonists still have motor capacity, but they don't like the heroin. How do we know that they don't like the heroin? Because the next day, they don't run for it. But this is the day they got the heroin. There's no haloperidol on board on this day. They get the heroin when they run for it. So what happens on day 16, the animals are getting the same stimuli. They're getting the reward. It's just like day 14. The only difference between day 16 and day 14 is that on day 16, they remember this crappy heroin injection they got the day before. Okay, brain stimulation is rewarding. You all know about this. Jim Olds discovered this with Peter Milner. They put a rat in an open field. Whenever he went over to this corner, they gave him electrical stimulation of the brain, and the animal kept going over to the corner. They figured out that the stimulation was rewarding. They were able to train rats to press a lever to get the stimulation. And the bottom line here is that dopamine antagonists block this reward as it blocked food reward and heroin reward. And again, it's a beautiful experiment done by one of my students, and I have the reference list for you if you want to see the details. But the bottom line is that dopamine antagonists block the rewarding effects of food, of heroin, of brain stimulation reward. And all of this has become irrelevant now because of the discovery by Boyden and Diceroth that you can optogenetically activate the dopamine system. And lots of people, some of them at this meeting, have already done this. And it's very clear that activating selectively the dopamine neurons in the brain is clearly rewarding. So activating the dopamine system is rewarding. what does burst firing have to do with it? Well, back to Tony Grace, here's burst firing. Burst discharges in 2 to 8 or 10 spikes, all linked to each other, each impulse a little weaker than the other. And these bursts are triggered by ionophoretic glutamate. They're not triggered by GABA. GABA doesn't make cells burst fire. GABA can increase or decrease the rate of firing of cells in pacemaker mode, but not this burst firing. It's never caused by GABA agonists or GABA antagonists. It is caused by glutamate. And Grace suggested that the burst firing and pacemaker firing have separate behavioral functions. This does one thing, and the other kind of firing does something different. His group, boy, I've got a lot hidden behind here. His group also showed that the pedunculopontine and lateral dorsal tegmental nuclei provide the glutamate input to the dopamine neuron that's capable of triggering this burst firing. How do we know this has anything to do with reward? We know that from Wolfram Schultz. Who showed that, can you still see my pointer now?
Yeah, we do, yeah.
Okay, so he showed that these dots represent the firing of dopamine cells. If an animal gets a reward, the cells burst fire following the reward, and you can see that in the histogram above it. But if the reward is predicted by a conditioned stimulus, then the burst firing occurs after the conditioned stimulus, not after the reward. And the animals learn this response to predictors after having this unconditioned response to the actual reward. They already know the response to actual rewards. They don't need dopamine to respond to. The dopamine system knows about reward because if there's a conditioned stimulus that says the dopamine is coming, and then you don't give the dopamine reward, the cells go silent. How does burst firing establish burst firing to predictors? But when an axon of cell A, this is Hebbs Theory written in 1949. Some of you weren't alive then. When an axon of cell A is near enough to excite cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency at exciting B is increased. Hebbs suggested that neuronal discharges increase a neuron's connectivity with input neurons that were active just prior to that stimulus. We tend to call these eligibility traces. These are reward predictors. And this theory offers a mechanism for how burst firing in response to unconditioned rewards causes burst firing to predictors that fired just before it. For those of you who haven't read Hebb's book, it's a long book. Many of you aren't going to read it. I want to suggest a shorter book. Hebb wrote a book called An Essay on Mind in 1980. It's only 150 pages long. It recapitulates what's in his longer book, and it's very well written. It's an excellent book, and I highly recommend it. This principle is also going to explain how reward-induced burst firing potentiates glutamate GABA long-term potentiation. Long-term potentiation was discovered by Bliss and Lomo. Bliss and Lomo discovered long-term potentiation. They discovered it in hippocampal granule cells. Repeated stimulation of glutamate input in the striatum to GABA output neurons produces long-term potentiation. And it happens in the striatum as well as in the hippocampus. How it works is best explained by Eric Kandel, who is working with long-term potentiation in aplesia. He showed that by blocking serotonin's function in aplesia, you could eliminate the long-term potentiation of glutamate input to GABA output. And in the mouse, he showed that the neuromodulator is dopamine. Dopamine does the same thing that aplesia is doing here. If you knock out dopamine, you don't get long-term potentiation. And Kandel explained this with Hebbs' model. So again, I advise you to read Hebbs' little book if you're not going to read Hebbs' big book. In any case, dopamine is critical for long-term potentiation and for long-term depression. Calabrese's group has shown that dopamine antagonists block formation of long-term depression, and long-term depression is restored by pretreatment with dopamine itself. And Calabrese, Kandel, and many other people have shown that long-term potentiation occurs in glutamate GABA synapses, depends on dopamine. How does that work? This is a study done by Reynolds, Highland, and Wickens in the rat. The rat has this brain. It has a cortical neuron projecting to the striatum, synapsing on GABA output neurons that are related to motor function. And it has a dopamine synaptic input to that synapse. And what they did was first train rats to lever press for repeated stimulation of the substantia nigra. Once the animals had learned that, they anesthetized the rats and they did and they showed that repeated stimulation of the glutamate pathway would potentiate the connection to the GABA pathway. If the dopamine system and the glutamate system are activated at the same time. So if you stimulate the substantia nigra in conjunction with activation of the input to the GABA neurons, you get long-term potentiation. So I've lost the track of thought here, but two things to remember about burst firing. One is that reward-induced dopamine burst firing caused by the reward itself enables burst firing in response to a predictor. That happens in the dopamine system. In addition to that, reward-induced burst firing enables long-term potentiation in sensory synapses on motor-related output neurons. This happens outside the dopamine system, between glutamate and GABA cells at the tip of the dopamine synapse. Okay, another thing to remember about glutamate GABA LTP is that burst firing of dopamine enables responses to reward predictors or to punishment predictors. It doesn't tell you whether you want to respond, but it stamps in the connections between the glutamate stimulus and the GABA response pathway. A hungry animal may want to respond, but a sated animal will not want to respond. And wanting to respond is motivational function, and it's related to current behavior, and it's coming up now. So pacemaker firing, the other form of activation, forms motivation. Now I really can't see. I got rid of you. Okay. Motivation is a state of readiness to react to stimuli. And it's a readiness to react to a certain class of stimuli. And that class of stimuli is predictive stimuli. Stimuli that predict rewards and stimuli that predict punishment. Motivation is a state of readiness to respond to those stimuli. Okay, pacemaker firing controls motivation. Pacemaker firing occurs spontaneously The cells here are firing 2 to 4 times slower than they respond in bursts. It's generated by a slow depolarizing intracellular current. And the rate of discharge is modified by GABA or by GABA antagonists, but not by glutamate. So GABA makes them fire slowly like this, and GABA antagonists disinhibit them to fire like this. And again, Grace suggested that burst firing and pacemaker firing have separate behavioral functions. So I want to talk now about pacemaker firing.
Roy, there's about 5 minutes left to the time, just to let you know.
I'm sorry?
There's about 5 minutes left in the time.
Wow, I'm in big trouble. Everett and Robbins have a 2-lever task where animals can where animals press on lever A in order to get access to lever B. When they press on lever A, they get access to lever B. When they press on lever B, they get a reward. So pressing on lever A is a measure of motivation. And if you block D3 receptors, you attenuate responding on lever A. Several groups have argued that motivation is affected by dopamine. Ikamoto showed that dopamine antagonists or VTA GABA decrease running speed but don't affect eating. Salamone showed that VTA striatal damage affects high ratio responding without affecting free feeding. The animals eat the food, but they just don't work as hard for it. Berridge has shown that amphetamine and amphetamine-induced sensitization will increase the willingness of animals to lever press for reward. So variation in dopamine levels cause variations in responding to predictors, motivation. Another thing that will control motivation is hormones that act directly on the dopamine system. Yu has shown that leptin will depress lever pressing for cues that predict a cocaine reward, but they still lever press normally for the reward. Okay, I'm going to talk very quickly about addictive drugs. Bozarth and I suggested in that 1987 paper that all addictive drugs activate the dopamine system. Susie Imparato and Gigi DiCera have shown that for amphetamine, cocaine, morphine, methadone, ethanol, nicotine, barbiturates, they all do elevate dopamine levels. These are experimenter-selected doses, but people have shown that with doses that the animal selects, you can get elevations, at least with cocaine, amphetamine, and opiates. Dopamine antagonists make these rewards ineffective. Again, this is a study that's explained in some detail in the references, but I'm not going to talk about it here. Opiates are rewarding in proportion to their ability to elevate dopamine level. This is a low dose of Damgo, a mu agonist of dopamine receptors. This is a higher dose of DPDPE, a delta agonist. They have the same effects on behavior. This is lever pressing for reward. And they have equal effects on dopamine. And finally, Michael Bozarth has simply shown that addictive drugs are rewarding when injected into the brain. So he showed that rats will work for lever press microinjections of morphine when directly injected into the ventral tegmental area of the brain. Those injections don't make animals dependent, and the animal doesn't lever press for injections into other sites of the brain. And a variety of people have shown that other drugs are also rewarding when injected directly into the brain. So addictive drugs are clearly rewarded, and self-administered doses of some of them elevate dopamine strongly. Nicotine and alcohol, and THC and caffeine elevate dopamine levels at much lower levels. It'd be nice to know what self-administration doses do. It looks like caffeine may not activate the dopamine system by itself. It'd be nice to know if, for instance, they can stamp in long-term potentiation, as these other drugs do. I want to mention quickly George Koob, who has a long interest in the dark side of addiction, which means the inability of addicts to eliminate drug-taking habits. His work turns out to be very important for the history of CRF, corticotropin releasing factor, and it's very important for the search for addiction medications. CRF is critical for maintenance of addiction. We've got a nice study about it. Robert Wheeler has a very nice study about it, and those references are in my list. And here are my concluding points. Dopamine-free animals don't learn much, if anything. Dopamine burst firing stamps in learning. Dopamine pacemaker firing motivates action. Dopamine does both of these things. It stamps in learning and it motivates action. Thank you for your attention.
All right. Well, thank you, Roy. So just in time, I think the timing is perfect. So we actually have enough time for questions. So if anybody would like to ask questions, please go ahead. Meanwhile, I mean, I don't see questions myself on the Q&A window, but I do have a question myself. I would like to know if you can speculate on, you've talked about pacemaking driving some, basal tone of dopamine, and do you think there's any roles for dopamine in terms of the basal secretion, independent of firing, for which most neurons actually do as well? So there would be like probably two tones of dopamine. So a tone driven and maintained by spontaneous firing, pacemaking firing, and then some basal tone of dopamine independent of that.
Yeah, I don't know much about it. I think that basal tone is probably enough to flood D2 receptors, to activate D2 receptors, and so it probably does have a function. But I don't know much about what it would do.
Thank you. There's a couple of questions that came in. There's a question by Genoveva saying, Dr. Wise, thank you for the interesting lecture. You mentioned the history of elevations of dopamine levels by all drugs of abuse. Can you comment on the history of studying changes of dopamine receptors associated with drug abuse? So plasticity in the receptors in the context of drug abuse.
Well, we know that you can sensitize the system. D1 antagonists can interfere with drug seeking, and D2 antagonists can interfere with drug seeking. That seems to be related to the cooperativity between D1 and D2 function. But dopamine antagonists generally block the rewarding effects of the drugs that we've tested.
There's also a question by Min Jung mentioning, well, first, thank you for this dopamine history walkthrough. It's amazing. In your mind, what would be the next big question to ask about dopamine? So the one that I guess nobody thought about yet.
Well, the question that I've always wanted to ask about was the loading of D1 versus D2 receptors with the differential sensitivity of the receptors. That's been answered by Sabatini yesterday. The other question is about self-administration of the drugs and what effect they have on dopamine, and whether the effects that these other drugs that activate the dopamine system weakly activated sufficiently to cause the learning formation of long-term potentiation.
Thanks. There's a question by Amir asking, is it fair to say that most of the research has been on what dopamine controls and not much on what controls dopamine? Any specific reason?
Well, pacemaker firing controls dopamine, and rewards and punishers control dopamine. What more do you want?
Yeah, and there's a lot of inputs to dopamine neurons that people have studied over the years.
Well, certainly cholinergic neurons and glutamatergic neurons from pedunculopontine and lateral dorsal tegmental nucleus control the burst firing of the neurons, which stamps in learning. Pacemaker firing is controlled by internal degradation.
And we learned a lot during this meeting about the local control of dopamine axons as well.
Yeah, I don't know. And I know that there are fluctuations of nicotine in the striatum, in the nucleus accumbens, that can modulate dopamine release, but can also modulate burst firing within dopamine axons. And I don't know anything about its function and behavior.
There's a question by Christopher asking, what's your take on the function of D3 receptors, both pre and post-synaptically? Are they always saturated?
I don't know. The interesting thing about all these receptors is they often operate in heteromers. D3 receptors are co-localized. And I don't know much about what they do alone. I don't know what they do in heteromers. We can just kind of guess. I don't know.
These are tough questions. And there's a question by Andres asking about depolarization block of dopamine neurons and whether there's evidence that this occurs in behaving animals, and if so, what would be its roles, its physiological roles?
I don't know. You can produce depolarization block by repeated injections by experimenters, but I don't know if it occurs spontaneously. So I don't know what it does.
And then there's a question by Leticia asking the role of dopamine in is there a firing rate switch from safe use to risk use in the context of drug abuse?
I think that there's not. I think that if you cause burst firing in the dopamine system, it's going to stamp in learning. Your habits are going to get progressively stronger, and it's eventually going to become dangerous. Dopamine is really important for habit-forming effects of addictive drugs, as well as gambling, as well as video games and a variety of things. It's going to stamp in habits, and if it stamps in habits enough, it's going to disrupt the rest of your behavior.
And then there's Carl is reminding you to share your list of references that you mentioned. It's not there at this final slide, but perhaps you could set that to the meeting organizers, and they will be able to post it on the Slack channel of this section of the meeting. You could just do that. There it is. And maybe a final question, Roy, if you can bear one more question. There's a question by Anissa Abidargam asking, it just flipped. I just lost it on my list. Okay, I see it again. The data from Sabatini is not consistent with much of the human imaging data showing dopamine release can occupy D2 receptors, so Volkov and Abidargam and others, and that baseline occupancy, at least in healthy volunteers, is about 10%. Of course, D1 would be even less occupied, but the data would suggest that D2 are not fully occupied in humans. So what is your idea about that? Your thoughts?
I don't know. In any case, I think it should be the case that a decrease in dopamine firing is going to show up to a D2 receptor strongly, whereas an increase in dopamine release is going to show up to a D1 receptor strongly. In the rat, Sabatini's data suggested that a beautiful case that had only worked in one.
Okay, well, I think we'll stop the session now, and if there's some of the further questions that we missed, these will be sent over to Roy so that he can answer back, and they will be shared in the Slack channel.